Yeah, so hi, I'm Kavya. I'm part of Edward's group, the new group, and today I will be discussing some of my and my coworkers' work on the practical synthesis of novel fluorinated antivirals for the treatment of HIV/AIDS and COVID-19. Okay, so I'll give a short introduction to fluorinated small molecules and specifically antivirals, and then I'll go more in depth into the application of these fluorinated antivirals to treat HIV/AIDS, which is our NNRTI work, and to treat COVID-19, which is the carmofur work. Okay, so for the past century or so, organofluorine compounds, which are basically small molecules with a fluorine atom or a fluorinated functional group attached, have been attracting attention. And this interest in fluorinated small molecules started in 1954 when fludrocortisone, which is the first fluoropharmaceutical, was brought into market, and again in 1980 with the popularity of fluoroquinolones, which are fluorinated antibacterial agents. So with the success of fludrocortisone and fluoroquinolones, the number of fluoropharmaceuticals has increased greatly in the past 50 years. As of 2020, 300 fluoropharmaceuticals had been registered, and in 2019 alone, 43% of the registered pharmaceuticals that year were fluorinated, a majority of which were FDA approved, as shown in blue in this diagram. So the popularity of fluoropharmaceuticals is skyrocketing, which begs the question, why fluorine? Why not any other element? Well, let's review the properties of fluorine. Fluorine is a halogen. It has the greatest electronegativity of all elements. It forms strong bonds with carbon as compared to other atoms, such as hydrogen, due to the partial ionic character of the bond, and it's generally not present in natural products. And these properties lead to three advantages of fluorination. First is the fact that the replacement of a hydrogen for a fluorine, for example, the alteration of a CH3 to a CF3 would result in minimal alteration to the parent structure as a whole in a drug candidate, as hydrogen and fluorine are both small-sized atoms. The second is that, as we discussed, carbon-fluorine bonds are some of the strongest bonds that carbon can form. So fluorinating a compound can increase its metabolic stability, by fluorination. And the third is that since fluorine is the most electronegative element with an electronegativity of 3.98, the addition of a fluorine to a drug candidate induces bond polarization, resulting in the ability to alter the lipophilic hydrophilic balance of a compound. And with these three properties contributing to our interest in fluorine when producing synthetic and semi-synthetic drugs and compounds, our group has explored various fluorinated compounds, but the topic of my discussion today will be two specifically fluorinated antivirals, efavirenz, which is an NNRTI for the treatment of HIV/AIDS, and carmofur for the treatment of COVID-19. So I'll be going into the NNRTI work first. Okay, so first, before I go into the NNRTI work, is a brief discussion of HIV/AIDS, in case anyone needs a review. So human immunodeficiency virus affects approximately 37.7 million individuals worldwide, and when left untreated, it can lead to acquired immunodeficiency syndrome, or AIDS. HIV, basically the way it works, it infiltrates CD4 plus T lymphocytes, which are a type of white blood cell essential to the immune system. And inside that cell, it replicates itself, then kills the host cell. And basically when that CD4 plus count drops below a certain number, I believe it's 200 cells per millimeter cubed of blood, the HIV has officially progressed to AIDS. So a very important step that I didn't really touch on in the last slide, is step 3 that you see in this diagram, where HIV, or it's called reverse transcription, where HIV converts its viral RNA to DNA. And this step is done by the enzyme reverse transcriptase. And without this conversion, HIV is unable to replicate. A class of small molecules called NNRTIs, or non-nucleoside reverse transcriptase inhibitors, are basically meant to inhibit that reverse transcriptase, which aids in reverse transcription. They allosterically bind to and inhibit this enzyme, and this basically halts viral replication, preventing HIV from progressing to AIDS or progressing at all. Currently, there are 6 FDA-approved non-nucleoside reverse transcriptase inhibitors, and I might butcher their names, but it's nevirapine, delaverdine, efavirenz, etravirine, rilpivirine, and doravirine, the first 3 of which are first-generation NNRTIs, and the second 3 are second-generation NNRTIs. So, as I mentioned before, the focus of our work is efavirenz, which, as you can see, is a fluorinated small molecule. Yay! So efavirenz is one of those first-generation NNRTIs, and first-generation NNRTIs have been found to have a low genetic barrier, which means that there's only a single mutation required in its binding protein in order to confer resistance. And efavirenz in specific actually also has been found to cause central nervous system side effects, leading to many cases of efavirenz discontinuation. So while efavirenz was definitely valuable in HIV/AIDS treatment, the emergence of efavirenz-resistant variants of reverse transcriptase necessitates the continued discovery of novel NNRTIs to combat the evolving viral target of reverse transcriptase, which is exactly what we did. And this leads me into our computational work. So basically a general overview of the work. In order to produce more effective and potent NNRTIs than efavirenz, we designed analogs of efavirenz with various substituents, which I will go over in the next slide. But then we made those analogs, and then in order to produce the efavirenz-resistant variants of reverse transcriptase, we used homology modeling, which basically means inducing mutations in the protein sequence of reverse transcriptase to study the effects of those mutations on the binding affinity of its ligand, or efavirenz analogs. And then the third step, which you may have seen on the previous page, was to use molecular docking, where we docked our analogs to both reverse transcriptase and the variants we produced through homology modeling in order to get predicted binding affinities and see if any of our analogs produced or performed better than efavirenz in those efavirenz-resistant variants. And as I mentioned, we produced a library of analogs, and this is the library. Basically, we took the 5-position chlorine on the benzene ring of the efavirenz scaffold and rotated it around the 3, 4, 5, and 6 positions, and then did the same with a fluorine, methyl, and trifluoromethyl group. And basically, that was our R1 substituent. And then for our R2 substituent, we replaced the cyclopropyl fragment with various cycloalkanes, including cyclobutyl, cyclopentyl, cyclohexyl, as well as with phenyl, tertbutyl, and methyl fragments. And this resulted in a total of 112 analogs of efavirenz. And then, like I said, the next step was to homology model our variants. I think it's still loading, but in the slide, when it loads, we basically show, I believe, the Y188L mutant, where the 188th amino acid tyrosine is mutated to a leucine. Hey, David, are my slides lagging?
It's very slightly lagging, Kavya. I think why don't you exit the presentation and then just rescreen share?
Oh, no, there it is. It's there now. Well, okay. Yeah, that's what I was talking about. Basically, the 188th position normally is a tyrosine, and in the next picture, it's mutated to a leucine, and that's the Y1AAL mutant. And after our homology modeling, we docked our 112 analogs to the allosteric binding site of wild-type reverse transcriptase, as well as 5 single mutants and 5 double and triple mutants of reverse transcriptase. So you can basically see the binding poses of efavirenz and top analogs here to wild-type reverse transcriptase and all the variants. And basically, we found that many of our analogs did perform better than efavirenz in both wild type, as seen here, and in the variants in both single mutants and in double and triple mutants, as you can see the increased binding affinities. Okay. And here is a heat map representation of our results. Basically, warmer colors are better binding affinities. And as you can see, many of our analogs did perform very, very well in wild-type reverse transcriptase, the top one being analog 2P with a cyclobutyl and 6-trifluoromethyl substituent. And here are the heat maps for the single mutants. You can see the top analog for each in neon yellow. Basically, a couple trends. We found that our highest binding affinities were observed among 5th and 6th position substituents because the substituents at these positions maximized halogen pi interactions and hydrophobic interactions with the proximal binding pocket residues. And along with this, a clear trend also correlated binding affinities to the size of the R1 fragments in the mutants. For example, this was basically in the order of cyclopropyl, cyclobutyl, methyl, cyclopentyl, terpbutyl, cyclohexylphenyl, from the highest to the lowest binding affinities. And basically, this order kind of fits the relative sizes of the fragments, with smaller cycloalkanes having highest binding affinities and larger fragments having smaller affinities. And we saw that this might be a matter of steric factors, with larger fragments simply being too large for the binding pocket and resulting in diminished binding affinities. However, while this order and this trend was followed by wild-type reverse transcriptase and single variants K103N, Y188L, and Y188H, it varied in single mutants. It varied in the G190 single mutants and also in the double and triple mutants, likely due to those specific mutations in the pocket, as well as differences in the binding pose of the analogs within the pocket, which may have altered or limited the type of interactions that were possible. And yeah, this is just the heat maps for the double mutants. Okay, so now we had these great analogs that perform better than efavirenz in both wild-type reverse transcriptase and its variants. So our next step would be to synthesize these analogs. And a starting point to that would be synthesizing efavirenz itself. So here's the current synthesis of efavirenz. Now, there's a couple reasons why this current Merck synthesis is unfavorable. First is the fact that it's not atom efficient. Basically, it has a low conversion efficiency, or a low ratio between the mass of the desired product to the total mass of product, or total mass of products, yeah. And the second reason is that it uses an alkyl lithium reagent known as N-butyllithium or N-buly. The problem with this is that N-buly is pyrophoric and will ignite spontaneously on exposure to air, making the synthesis not only inefficient, but unsafe. Instead of using this reagent, we propose an alternate synthesis in which we use a chiral metal catalyst such as zinc, ytterbium, or indium triflate, and a ligand such as binap, binol, or silane in order to produce a safer, more practical, and more scalable synthesis. And this is currently a work in progress, so maybe in future colloquias, you'll see how that goes. And so that's all for NNRTI. So now I'm going to move into our work with carmofur, which is another fluorinated antiviral. So as we know, the current COVID-19 pandemic elicited a response from the scientific community, not only in the form of vaccines, but in the form of small molecule therapeutics for COVID. Many of these small molecules were not novel compounds, but rather they were drugs that were already on the shelf that were repurposed to treat COVID-19. So why exactly would we repurpose drugs as opposed to synthesizing drugs directly intended to treat COVID? Well, first of all, it's just faster to get existing compounds off the shelf just because they're already there. And the second is the ability to bypass regulations, since the drug would already be approved for whatever its initial target or application was, which in reality is also making it faster. Carmofur, which our project is focused on, is also one such repurposed drug. And I'm going to be talking about carmofur now. So yeah, carmofur is a 5-fluorouracil derivative, and it is originally an anti-neoplastic agent that is an inhibitor of acid ceramidase, which is an enzyme that is dysregulated in many cancers. Through drug repurposing screens, carmofur has been found to be a covalent inhibitor of the main protease of SARS-CoV-2, also known as Mpro. Mpro plays an essential role in the processing of polyproteins that are translated from viral RNA, which makes it an attractive drug target for COVID-19. Similar to reverse transcriptase, however, there are new mutations constantly arising in Mpro, necessitating the development of analogs of carmofur that outperform carmofur itself in these drug-resistant variants. In this computational work, alkyl amino acid and benzylic side chains were attached to the carmofur scaffold, producing a total of, I believe, 34 carmofur analogs, which were docked onto Mpro and its 51 variants to produce the results in the heat map shown. It was found that analog 2S with an amino acid substituent consistently produced high binding affinities in both wild type Mpro and its variants. We also pursued the synthesis of carmofur itself, which would then enable us to synthesize analogs of carmofur that may show better biological activity than carmofur. So the original synthesis of carmofur consisted of hexylamine and 5-fluorouracil, while our newer synthesis that was proposed instead uses 5-fluorouracil, since carmofur is a 5-fluorouracil derivative, and it uses an isocyanate instead. However, there is still one problem with the synthesis, and that problem, or inconvenience, I guess, was the solvent, which is pyridine. Unfortunately, pyridine is not only extremely toxic and environmentally problematic, but also consistently produced low yields of 3 to 35% in our synthesis. After rigorous solvent screens, however, we found an alternate solvent, which is NMP. Okay. NMP is less toxic and not as environmentally problematic, and is also easily removed by aqueous workup, thus creating an easier or more scalable synthesis. And it also has greater than 70% yields, which solves the yield problem that we faced with pyridine. Using our new conditions, we hope to synthesize 8 carmofur analogs using various isocyanates. Currently, we are working with 3 isocyanates: benzyl isocyanate, which is the top mechanism, 2-chloroethyl isocyanate, and transfer methyl cyclohexyl isocyanate. While we have optimized solvent conditions, we are yet to optimize conditions such as temperature and the presence of base for these reactions in the specific solvent, for which we are running test reactions enabled by 19F NMR. Using 19F NMR, we can perform NMR tube scale reactions and monitor reaction progression to efficiently optimize our analog reactions. And basically 19F NMR, it allows us to track the fluorine handles on 5-fluorouracil as seen in orange, as well as the carmofur analog produced as seen in purple. And this is perhaps another hidden benefit of having fluorinated compounds, at least from a synthetic standpoint. So yeah, that's all I have for my colloquium today. I would like to thank all my NNRTI and carmofur group members who I was not able to get all of them on there, but yeah, thank you to all those subgroup members, and also thanks Edward and ASDRP for funding and also an analysis because we used NMR spectroscopy. So yeah, thank you.
Awesome job, Kavya. That was an excellent presentation. That was a very good representation of how some of the things in our group take disciplines and areas of expertise from computer modeling, medchem, physical organic chemistry, all to do synthesis for unique applications in chemical biology. And certainly this space in antiviral research is one that is very, very, very much so important for not only the modern day, but also all the other diseases that are running around on Earth. So good job to Kavya. That was an excellent presentation. Any questions? Questions? Put the attendance form in the chat. Okay, that's the attendance form. Questions? Any questions? So Kavya, I think you scared all of them. There's no questions. Well, I have a question for you. What was the most difficult experiment or what was the most difficult thing to learn how to do, and what was the most difficult thing to actually do? And are those the same?
I think like within the stuff I discussed today, I'm assuming.
Well, yeah, that or any of the other stuff that you work on, because you work on a lot of things.
Okay, well, I'm going to talk about carmofur. I think like optimizing reactions, at least for me, is kind of, I guess that's the hardest thing to do, just because like it's a lot of factors that have to align. Like I said, although we have like our solvent down, I've been working with this 2-chloroethyl reaction for a while, and you know, it's always one thing or the other, but you feel really great when you finally optimize it, or so I'm assuming. I hope to get there soon. And I guess the hardest thing to learn to do is also kind of related to that. It's just like you have to stick to it, I guess, because chemistry is in slushy era. Yeah, we make slushies. But yeah, chemistry, it takes a lot of effort, and you just have to stick to it because it's not 99% of the time, it's not going to work out the first try.
Yeah, I remember when Kavya first joined the group, she had some research experience from some summer program, right? So she came in with the whole presentation, goes like, oh, well, this is how we're going to treat cancer. Like, so, and then she brought to that meeting a chemical, the results of a preliminary small molecule drug. And then I asked her to draw the structure. Do you remember that? And there wasn't a lot of drawing that happened after that, for at least those few minutes after. But good job. You've really risen to the top of a lot of these things, and certainly have gained a lot of skills. And not only being able to make molecules, but also make molecules with meaning and use NMR to make molecules with meaning. That to me is just awesome. So cool. All right. And I do have one suggestion for you, Kavya. And I think if you want your Zoom to lag less, have fewer tabs. Oh, you have a question in the chat window.
How did you guys decide on using carmofur to treat COVID? Were there other molecules that you are interested in working on? I'm actually not sure. Like I said, carmofur is just one of those repurposed drugs, so maybe they just picked it, but this effort was started long before I was here. So I'm not really sure, but yeah. Ed, thoughts?
Yeah, so that is and we'll answer the next question in a second, but so back in 2020, when the pandemic just began, things got really scary really quickly around March. But around December, if you all remember December 2019, it was less scary and more like, what is this weird disease that's going around in Asia? And for a medicinal chemist, that was an exciting time. That was a very exciting time. It was like, oh, there's a new disease, let's cure it. And so we started looking into the proteins that were involved. And one of the early targets was the main protease, because this has a lot of sequence similarity with existing coronaviruses out there. And in fact, the first X-ray structures of the protein that we're targeting here was published in, I believe, November of 2019. So just a few months into the first identified case of COVID-19 in China. And so we began at that point in time in our group, before COVID even hit the US, we began an effort to identify small molecule agents that might be effective in targeting this particular protein. And in fact, we published a paper back in 2020. I can put it in the chat window. Yeah, so this is the paper that we published in 2020. Where is it? Here we go. All right, so I'll put it in the chat window. You can all read it when you have time. Where we use high throughput virtual screening to identify these peptidomimetic small molecules that would, in theory, mimic the natural substrate and inhibit the enzyme. And so that was back in 2020. So we finished that effort, and then we got bored. And around the same time that we published this paper, about a week and a half later, another paper came out in Nature from a Chinese group identifying carmofur as a small molecule that covalently inhibits SARS-CoV-2. I'm like, oh, that's pretty cool. And it turns out it has a fluorine. And during that time in the group, there were some people in the group who were obsessed with fluorine NMR because it was so beautiful. So the early carmofur crew, Carissa, who's now at Johns Hopkins, Jesslyn, who's at Santa Barbara, Julia, who's at Stanford, who else? Neha, who's now at UCLA, those folks, and Udbov at UCLA too, began asking the question, well, can we use fluorine NMR to make these sorts of antivirals to treat COVID-19? It's like NMR plus synthesis plus chembio to treat a viral disease that we're all freaking out about here in the United States. And so that's exactly what we did. And it took us about 1.5 years to make carmofur on scale in reasonably good yields, thanks to the people like Kavya who helped to troubleshoot this reaction. So that's how we got involved in carmofur. It was like a series of serendipitous events. Okay. Other questions?
Could you repeat again how exactly carmofur combats COVID-19? Okay, yeah, so I think I just mentioned this again, but basically it binds to, let me go back to that slide. Please don't lag. It binds to COVID-19 Mpro, and it, which like Ed said, is one of those really important proteins in COVID, and basically just inhibits that protein, I guess kind of stops the function.
Other questions? So carmofur, for all of your benefit, is several $100s for a few milligrams, and Zena and Kavya and others in the group, we've produced maybe about $500,000 worth of carmofur in a few sittings. So that's pretty good. Yeah. Okay, great. So there's no other questions. Great job, Kavya. That was a pretty chunky presentation. See you all next week. Next week we have a presenter from Dr. McMahon's group and then another one from my group. And then maybe we'll hear a biology one. Let's see. Have a good night, folks. Goodbye.